morning dear students and welcome to Genesis. In today's lecture I am mainly going to cover two experiments. One in which we prove that chlorophyll is necessary for photosynthesis, and second to prove that light is necessary for photosynthesis. But before we know about these experiments, it's important for us to know how to de-starch a plant. De-starching is that process where the leaves of a plant lose starch. And if there is any starch in the body, it will be found in the storage organs of the plant, but not in the leaves. How can we do this? By simply keeping a plant in shade or in a place where there is no sunlight for 24 to 48 hours. After this, there is an experimental process which you perform, and you can see that actually the leaves lose plant if you keep them in shade for 24 to 48 hours. And that is mentioned here. This is the test of a leaf for starch. If you perform this, you will find that the leaves show no starch if they are kept in shade for that stipulated period of time. So imagine you took a potted plant, you kept it in shade for that period of time, and then you have plucked a leaf from that plant and you are performing some set of steps to check if that leaf has starch or not. What do we do? First, what do we do is we take the leaf and we dip the leaf in boiling water for a minute. First question, why are we putting the leaf in boiling water? Because we want to kill the cells and we want to make sure that the cell wall is damaged, because there are some other steps after this which requires damaging of the cell wall. Right after that, you're going to take out the leaf and you're going to put it in a methylated spirit over water bath. There can be two questions that can come to your mind here. Why suddenly are we using methylated spirit? Leaves are green in color, and as we all know, there is a test for starch which is called the iodine test. This we have done in our junior classes also, where what do? We take a sample and we put few drops of iodine over it. Iodine's original color is brown, but if it comes in contact with anything which contains starch, it turns blue-black. So for example, you take a peeled potato and put few drops of iodine over it, you will see that the iodine which was in the jar was brown in color, but the moment it comes in contact with the white base of the peeled potato, it shows a color change of blue-black. So here, if we put iodine on green leaves, the color change will not be evident, because green itself is a very dark color, so you will not be able to understand the change. So what we want to ensure is that the leaves or the leaf that you are using should lose its green color. And how will it lose its green color? Only when it loses chlorophyll. How can it lose chlorophyll? Not by boiling in water. If you boil in water, the leaves will still remain green in color. Otherwise, like when we make vegetables, when we make spinach at home, the spinach never loses green color when you boil it and cook it, right? Because chlorophyll is not soluble in water. Chlorophyll is soluble only in alcohol, and that is why we are using methylated spirit here. The moment you put the leaf in a test tube which contains methylated spirit and you keep the setup in a water bath, that means a beaker that has water, and you put flame, you are going to observe with time the leaf is going to lose its green color, and the water and the methylated spirit will turn green in color. But the second question that we can have mind is, why am I not directly heating the test tube? Why am I keeping it in a water bath? Because methylated spirit, alcohol, they are inflammable, so the entire test tube will catch fire. That is why you cannot put them in direct contact to heat. You have to keep it in water bath. Once you are done with this step, what you are left with is a white colored leaf which is very brittle right now. So what do? You take out the leaf and place it again in hot water. Now why am I doing it? To make it soft again. And the last step of this entire process is you take that leaf and you spread it on a dish. That means you keep it on a dish and pour iodine solution over it. What are you expected to see here? Will there be a color change or will there be no color change? If the leaf has no starch, that means it has been absolutely de-starched, there will be no color change. The leaf, the color of the iodine will remain brown and there will be no color change. But if the leaf has starch, then of course it will show a color change. The iodine will change from brown color to blue-black. Now, since before I started explaining this, I have already told you we have kept the pot in shade for 24 to 48 hours. That means we have ensured that there was no photosynthesis happening. So whatever little bit starch was there in the leaves were already consumed or were kept in the storage parts of the plant. So there is no starch in the leaf. And if you take such a leaf and you do these processes, and after that you come to the last step, expected to see no change in the color of the iodine solution, hence prove that the leaf or the plant has been de-starched completely. Now we are going to take this potted plant and perform the other two experiments on it. Okay, so let's quickly understand the next experiment. In the first experiment, we are trying to prove that chlorophyll is necessary for the process of photosynthesis. If you remember, we know there are 4 essential conditions for photosynthesis. One is light, which is sunlight. Another is chlorophyll pigment itself. Another is carbon dioxide and water. So we are testing one such parameter, which is presence of chlorophyll. Now for this experiment, you need some special kind of plants such as croton or geranium. You also have to remember these names because these are given in our syllabus, and coleus. All these names are special because these plants are also known to have variegated leaves. Now let us understand what are variegated leaves. So variegated leaves look like this. That means they are not completely green like any other leaf that we see around, which are completely green. They are not completely green. So they will have patches of other colors as well. Like they may have patches of white. They may also have patches of red and green. So they are not completely green. Why are we doing this? We are doing this because we know in such leaves the concentration of chlorophyll pigment is lesser in those areas which are red or white in color compared to rest of the area. Like for example, if I ask you that if a plant which has red leaves, there are many such plants which have red leaves instead of green, are they photosynthesizing or they are not photosynthesizing? They are photosynthesizing, of course, but then what about their chlorophyll pigment? They have chlorophyll pigment, just that the quantity of chlorophyll pigment in their leaf is lesser compared to the other colored pigments. So in such variegated leaves, you are going to see that it's not a completely green leaf. It will have areas of higher chlorophyll concentration and low chlorophyll concentration. So what we do is we are going to take such a plant and we are going to de-starch the plant first. That means we'll keep it there, keep the potted plant in shade for 24 to 48 hours. After that we take it out, and we already know that this is the span needed to de-starch the leaves. So we are sure now that the leaves will not have any starch. Now we take it and keep it out in the sun. Why are we doing that? Because we want to ensure that whatever small period of time for which we are going to keep this potted plant out in the sun during our experiment, only that is the time when the plant is allowed to photosynthesize and produce starch in its leaves. Whatever starch was already prepared, was already there in the leaves, we have removed it by the process of de-starching. So now after that, so you kept it out in the sun and now you pluck one leaf, perform starch test on it. But before you perform the starch test, you are going to use a tracing paper and you are going to take the prints of all those areas. Say for here, these are green areas and this is the non-green area. So you are going to use a tracing paper and trace the leaf and mark the areas which are green. And now once you are done with that, you are going to perform those steps which I explained. Like you are going to first boil the leaf in water, then you are going to boil it in methylated spirit, again put it in hot water, and then finally smear iodine over it. Once you do that, what are you going to observe? You will observe only the areas that were green in color. That means those areas which had chlorophyll give positive iodine test. And what do we mean by positive iodine test? That means here there will be a change in color of the iodine from brown to blue, black. What about the non-green areas? They are going to give negative iodine test. That means there will be no change in color. Why is this? Because these areas which are green in color, that means they contain chlorophyll pigment. Since they contain chlorophyll pigment, when the potted plant was kept out in the sun, they could photosynthesize and produce starch. Whereas the other areas, they do not contain chlorophyll pigment. That is why they did not photosynthesize and there was no concept of producing starch. Now when you do the iodine test, you can see the green area will give a positive iodine test, where when it comes in contact with starch, the iodine changes color from brown to blue black, whereas the areas which were non-green in color did not produce any starch, so the iodine did not show any change in color, it remained brown. Now let's do the next experiment. Now in the second experiment, we are trying to prove the other parameter which is essential for photosynthesis, and that is sunlight. Now in this experiment, you will take a potted plant. It need not be a variegated plant, just a basic potted plant, and you are going to keep it out in the shade for 24 to 48 hours. Why are we doing this? We are going to de-starch it. But now before you keep the potted plant back into the sun, you are going to do one thing. You are going to take one big leaf, let it remain attached to the parent plant, but cover a part of the leaf with a black piece of paper. But you are also going to make a small cut in the black piece of paper. It can be of any shape, a star, a circle, however, and then you are going to keep it out in the sun. Why are you doing this? Because you want to ensure that when sun rays will fall on the leaves, they will access the cut out part, but not the parts which are covered with the black paper. Let it be in the sun for few hours, and now you pluck the leaf and perform iodine test. That means all those steps that I have already mentioned, we are going to perform all the steps together. Once we do that, we are going to see the part of the leaf which was covered with the black paper. So imagine this is the part that was covered with the black paper. This part will not give you positive iodine test, hence it will give negative iodine test. Negative iodine test means what? There will be no color change of iodine. That means this part will remain brown in color. Why? Because since it was covered by the black paper, sun rays could not reach this area and there was no photosynthesis performed here and no starch produced. As a result of which, when you did a starch test, you got a negative iodine test or negative starch test, where there was no color change of iodine. It remained brown in color. This part which was exposed to the sunlight gave positive iodine, or you can also call it starched test. What do we mean by positive iodine test? That means there was change in color. That means the color here showed blue-black. So iodine that was poured here remained brown in color, but the iodine which was poured here changed from brown to blue-black, which clearly tells us that in absence of sunlight, the plants do not produce starch because they do not photosynthesize. So that's all for today's lecture. I'm going to meet you soon in my next video. Do not forget to like and subscribe my channel. Thank you.